in this presentation, what I'll be covering is, well, the secret life of seeds. We really don't know much about them other than what we see, right? Well, this presentation will cover what goes on under that seed coat. All right, here we go. Well, pretty much, you get a seed. You buy your seeds from the store or you harvest them from a plant and you just look at it and you think, well, I just need to add water. And then they magically germinate, usually or hopefully, right? But what we don't know is that there's a whole lot of things going on in the life of the seed. Once you add water, it sets off a whole different reaction within those seeds. And even before we add water, there's a whole secret life that's going on inside that seed. But to make germination happen, we have to provide the right environmental conditions prior to us receiving those seeds and then after we put them in the ground. As I said, most of the time, well, before I get into the rest of the presentation, this presentation will cover the point when the seed is mature, almost mature, to the point that those first leaves, the cotyledons, are coming out of the ground. That is part of the life cycle of the seed that we will be covering. Now, when you get a seed and you look at it in your hand, you've noticed that seeds all look different, particularly if, say, you're working with prairie seeds, all those different flower seeds that need to be planted in the prairies. You can see that they're all sorts of different colors. They're beautiful. They have so many different textures. And if you've ever tried to crack a sunflower seed open or a peanut seed open, you'll see that their seed coats vary in thickness, in hardness. And you'll notice that seeds also vary in their characteristics. They have this outer harder coat, but then on the inside there's a thinner coat. And these coats are very important. They all serve their purposes. And really what they're doing is protecting from desiccation and pests and some other things. These seed coats are vital. And typically that's the only part of the seed that we recognize. And on the inside, as you've broken open that peanut or that sunflower seed, you find the true seed, the mature ovule. And in that ovule is everything that little seed needs, or almost everything that little seed needs to grow into a plant. Inside you have the cotyledons, the first leaves. You have the plumule and the hypocotyl and the radicle. And these are all words that you really don't need to know, but they're just the technical words of what's inside that seed. And then there's, well, the cotyledons, and that's what we see when the seed finally germinates. Actually, you may see the cotyledons or you may not, depending on if they come in above ground or below ground. And the cotyledons are where a lot of the energy of the plant is stored. That is where most of the food for the plant is stored, but not in all cases. And in the plant, or in the, not the plant, but the seed, there's all sorts of different things stored in there. We have our carbohydrates and proteins and also our lipids, but they're not all in the same ratio. Every seed is different. Every seed has its own niche. They either grow in really cold places or tropical places or really dry places or really wet places, and they can grow in the tundra. And so based on all these environmental conditions that these seeds grow in, they have different amount of carbohydrates, different amount of proteins and lipids. And these aren't just one type of carbohydrates. In carbohydrates, there's the sucrose, the trehalose, hemicellulose, and these all vary in different amounts of starches and fiber. They all have their role, whether it's structural or to provide energy. And in proteins, proteins are stored in different places in the mitochondria, in the cytosol, in the protoplastids, in the nucleus. But then they're also stored as amino acids, and those eventually turn into different things. They're building blocks. And if you look at this picture here, you can see that, for example, chestnuts or oak trees or oak seeds, they have very, very high carbohydrate load, but then a pine has a very low carbohydrate load. But a pine has a much more fatty and protein load on the inside. Whereas if we look at that chestnut seed, it had very high carbohydrate load, but then a very low fat load and a very low protein load. And so you can see seeds vary very much depending on what they're adapted to. Are they deciduous? Are they pine? or needle-leaved, they all vary. And when it comes to lipids, there are so many different kinds of fats inside a plant. And just like within us, we have so many different types of lipids within our bodies too. And these fatty acids, there may be fatty acids, which are building blocks for other parts, just like amino acids. But then they also have a lot of lipids that are involved in cell walls, in the bilayers, or the different parts of the cells. And so depending on what kind of seed you have, it's going to have different composition. But that also helps it in its later on life strategy on how it's going to survive and make it to germination and through the environmental conditions. There's other constituents inside of a mature seed. There's minerals, there's the nucleic acids, there's pigments, vitamins, and all sorts of other things that are all building blocks. They're all very important. And we don't think about these things inside the seeds. And how do they get there? Are all seeds getting all their stuff directly from the parent or the environment? Well, it depends. Some of the things that we find inside of the seed are genetically determined because, well, life history, but not everything. You're going to have your parent trees are going to have slightly different genotypes and phenotypes. And so depending on seeds that you get from down here or the same exact tree that you get in Illinois in a cold area, the seed composition inside will be different because of what they've had to endure living in the north and the south part of the country. But then also that parent tree, was it on poor soil? Was it on very good soil? That will also put into the seed the minerals, the vitamins, the amount of nutrition that's in the ground will also go into the seed. So it's dependent on how well are the growing conditions of the tree, the original parent tree. Now, once the seed is mature, it will go into a process called desiccation. So think about the seed on the tree or the plant in the flower, and it's green, and then it starts to darken up and it starts to get dry and hard, and that's the desiccation process. And that is very important. That is a pinnacle thing that has to happen. When the mature embryo has everything it needs and it dries out, it will have a very low percentage of water in the seed. Well, relatively. Some seeds go so dry that they're considered, or all seeds are considered anhydrobiotes or anhydrobiotes. And these are life forms that can dry out and desiccate, and then when you add water, they come back to life. So tardigrades are one of these forms, and there's other creatures, but I think that's fascinating that they are something that just can desiccate and then come back to life. And depending on how much desiccation that seed goes through will determine how long it can go into storage. And so seeds that keep a high water concentrate in them, a good example is oak seeds, they hold a lot of water still inside them. They don't store very well. They only store for about 2 to 3 months, and that's it. If you don't plant them or if they don't get the right conditions, they go bad. But something like Arctic tundra lupine seeds, they can be in frozen still and dehydrated for 10,000 years. And then once they get the right amount of water, the right conditions, they germinate instantly, which is incredible. In the pine industry, which forestry is very much concerned with, they can hold seeds if they're in the right storage conditions and desiccated. They can be in 50 years of cold storage and then still come back and have 66 to 25% viability. You know, you want to use them sooner than that. The Bastrop pines, a good example is this, is the Bastrop pines that were lost during the forest fires of 2011 at Bastrop State Park. The seeds were stored in a refrigerator 20 years ago or something like that. It was a while back. And that fire happened, and it just so happened at Berkshire Brothers, there were some seeds being stored in a freezer. And they were able to take those seeds and repopulate the Bastrop pines with the same genetic seed that was there originally. So if seeds are stored properly, we can bring them back and use them as needed. Now, when we talk about desiccation of a seed, there's two different kinds of desiccation. There's recalcitrant and orthodox. And I had hard time remembering what recalcitrant meant. And it pretty much means unruly. An unruly child, an unruly person. Well, recalcitrant seeds are unruly seeds. They do not want to go into storage. They lose their vitality very quickly if you try and store them. And that's because they have to keep a high moisture content. And so this includes mango seeds, oak seeds, walnut, those meaty seeds. They just do not like to dry. And if they do, they just lose their viability, even within a few weeks. So these seeds need to be pretty much planted immediately or stored very properly for a very short amount of time. Orthodox seeds are more the seeds that you think of when you're planting a garden. They can be stored for a year. They have an expiration date of about a year or two. They store very well. As long as you keep them in dry conditions in a paper bag, they will do just fine stored in the dark. Their moisture content will stay low, and then we just give them the right conditions and they pop right back. That's conservative, an orthodox seed. And then we have, let's talk about vigor, because the way we store seeds will affect how their vigor is. A seed that has high vigor is a rapidly growing seed. And these characteristics of vigor are influenced by heredity, how the seed developed inside. Was it under good conditions, or was the parent tree stressed while that seed was developing? How was it harvested, and how were those storage conditions? And then, of course, once you plant that seed, it may be a vigorous seed, but if you plant it in poor conditions, well, then it's not going to do very well. It's going to have low vigor. And then a seed with declined vigor is a seed that has aged. And what happens with those seeds, you can tell that these are seeds of low vigor. You plant them and they just don't germinate. And that's if they've been stored properly. But they just don't germinate. They are more susceptible to be attacked by microorganisms or pests. When they do germinate, they have very short radicals. They don't grow nice and perky. They just kind of, and their cotyledons don't emerge from the seed coat. And those are those first leaves. They just kind of stay stunted and they eventually just die. And what's going on in a seed with low vigor is there's been a loss of enzyme activity, a loss of the membrane integrity of the seed coats. And then this brings on all these other failures. And the enzymes just don't work. Things aren't happening properly in the seed. And we'll talk more about those proper things that have to happen for that seed to germinate well. But if the seed has been stored properly and it's desiccated, then it goes into a state of dormancy. And that is what a seed needs. It's like it needs a time to just sleep and rest. And it's a period of arrested growth, but there are still metabolic processes happening in the background, but they're very small processes. It's called after ripening. And this has to happen. And some seeds don't need this, but some seeds absolutely need this. And so in dormancy, you can plant a seed, but it's not going to grow. It's not going to grow until it's ready. And even if it's ready, if the ideal conditions aren't there, it's not going to grow. And so dormancy is this time when the seed just needs to wait it out. Either it's finishing its maturation process, maybe it has an extended long maturation process, or maybe it just is waiting for those optimal conditions. It's waiting for those external factors to take hold. And there's an advantage to this. Think about it. If you just fall off your tree or your plant, is it the right time to grow? Maybe it's really hot outside. It's really dry. There's not enough water in the ground to keep you alive. And plus, you're going to get pretty sunburned if you're a poor little plant coming out of the ground and you're not ready to endure that. You haven't matured as a plant. Also, the seeds may drop and it might suddenly get cold. And then you've got injury from freezing. And some trees can recover, some plants can recover, but not all. Some plants, once they freeze, that's it. So they have to wait it out. In the desert, you actually need the monsoon season to happen. And after an extended monsoon season, and the seeds can sense that. Once the right amount of monsoon season has happened and there's enough moisture in the ground, then the seeds germinate. And so the seeds have to go through this dormancy of internal and external factors so that things are just right. Now, the disadvantages of maturing and all of this dormancy process is that if you're a nursery and you want to grow a lot of trees instantly or a lot of plants instantly, you're going to have to wait all of this out, and you're going to have to figure out, how am I going to get all of these seeds to germinate exactly at the right time? Because you may have an order of 80,000 plants that you need to deliver in 4 months, and they have to be at exactly this height or this height for the boxes. And so nurseries have studied this extensively. Forest nurseries, plant nurseries, they all know what they're doing because, well, this is their bread and butter. Now let's talk about those internal factors that cause dormancy. Well, look at the seed coat. Bonus to you guys who can name a whole lot of these seeds. I mean, I see carrot, dill, fennel, chard. I see some seeds that remind me of prairie seeds, some native seeds that I've planted before for flowers. Just look at all those different seed coats. Some of them are very flat, some of them are round, some of them are pitted, and they all have their different purposes. Primarily, the point of the seed coat is to keep water from getting into that seed. Water, it has to stay out of the seed. It must stay desiccated until things are just right. Also, it's keeping out gases. Got to keep the gases out of those seeds. And then also, and by gases, I mean oxygen, but then other gases as well that we'll talk about. And then also that seed coat is a belt. It's a mechanical belt keeping everything inside. So even if things wanted to pop out, it's saying, nope, you're going to stay in there until this seed coat has been worn down and things are just right for you to open. And also this is very good for acorns because this helps them delay this is a good example. Acorns are delayed. They've got that pericarp, that hard outer shell, and that mechanical strength keeps everything inside. And it's got chemicals inside that seed coat that are saying, nope, we're not going to let reactions happen. This chemical is going to keep this inhibited until things are ready. And that seed coat also slowly lets water in at a rate that seed coat deems allowable, because remember, it's only good for 2 months, 3 months. And so that seed coat is keeping everything in check. Let's look at also what else causes dormancy. Genetics. That seed may need extra time to after ripen, as I mentioned before, and some seeds actually need 2 years to after ripen. For example, European ash, which is very different from our green ash from here, but it actually needs 2 years of, once it falls off the tree, that seed needs to go 1 full year and then all the way back around so that it will germinate in the spring of the 2nd year. Red oaks also do the same thing, but they stay on that tree for 2 years and then they finally fall. And then they'll go through a winter of over-ripening and stratification. We'll talk about that word stratification in a little bit. And then they'll finally germinate. Whereas other plants, they just fall off the tree and then suddenly germinate. It's very different. Then there's also, I just mentioned physiological dormancy, where, for example, those European ash seeds, some need 2 years, but then ours here in this part of Texas, our green ash, they have a dormancy that they can fall off the tree, and if everything's right, they automatically form a new seed, a new plant. But if conditions aren't right, that seed, our green ash, will actually go into a period of dormancy over the winter, and then pop up in spring. I grow trees for replanting projects for Master Naturalists. I plant about 500 trees every year as my main project, and I can go ahead and collect green ash seeds and experiment with this. And I go ahead and put them into a stratification process over the winter. But if I was really trying, I could probably grow some as soon as they fall off the tree in around, September, I think. I can't remember. But, I can time when I want them to germinate based on their physiology. And then there's double dormancy. And for example, with roses, there's the thick shell that keeps that rose from germinating but then it also has gases that keep that shell from also letting that shell produces gases that keep that rose from germinating. And so, you know, there's various ways that dormancy happens and various lengths. And that's just because every plant has its niche. If everything did everything at the same time, plants wouldn't be able to compete with each other and find their life roles as to where they will be on our landscape. Now, another thing that causes dormancy is hormones. There's abscisic acid, which is a growth inhibitor, but then there's gibberellin, which is a growth promoter. And then there's other growth promoters. Ethylene, for example, if you take avocados and you stick them in a paper bag or wrap them in newspaper, you know, you go to Walmart and they're really hard. Wrap them in newspaper, give them 2 or 3 days. The more you wrap together, the faster it'll happen. But the ethylene that those fruits are producing is the gas that will cause those avocados to ripen. And that's a hormone. And we can also do this with, I believe, carbon monoxide with tomatoes. You know, we can figure out how to make these plants ripen faster if we want, if that's what you want. Now, people used to think that abscisic acid and gibberellin had this ratio that they worked in. As one went up, one went down, and the other way. But actually, that's not true. And what we figured out is that gibberellic acid is actually dependent on light. And we'll talk about that later. But there's this whole thing going on in here in every seed. You know, you've got water, you've got your hormones, you've got your proteins, you've got your enzymes, you've got your sugars. And there's just a whole lot going in here. And hormones play a very big part of that. Now this is a point where we can take questions. Does anyone have any questions?
I don't see any questions in chat right now. I think it's because you are so amazing. We're just hanging on your every word. It's wonderful. Thank you.
A lot going on here. I recognize that. And I'm trying to make sure to get all of this information out, but trying not to go so high up here like my PhD classes where I actually get nervous because I feel like, wow, you know, all these forestry students know so much more than I do because I didn't grow up with a background of forestry or an education of forestry. So trying to bring it to a level that's challenging, but still where we can all learn something. And there's just so much. Fascinating.
One question, Laurie. You talked about the seed's ability to resist when there's too much water or too little water. And does that mean after, say, Hurricane Harvey, the seeds would be able to stay viable even though there was way too much water and under normal circumstances you would think they would just rot?
Well, Hurricane Harvey happened in August, was it? It was August. And at this point, we're usually in a very severe drought phase. Now, what's really interesting about Harvey, some of the effects that we saw after Harvey was that there was so much water, the seeds moved. And this also happened during Hurricane Ike. And so some seeds can float and move, and then some seeds can't float and some seeds can't move. And so some of them probably got saturated and did not do well, but then for some, it probably actually increased where they could go. And we'll talk about that a little more in this next couple slides or so.
Okay, and while we were talking, another question came in too.
Sure.
Lisa, would you please unmute yourself and ask your question?
Hi, good morning. You mentioned earlier about saving seeds, say that we didn't plant all of our seed for one season. You mentioned putting them in a paper bag and storing them in a dark place. I had
cool, dark place.
Yeah, don't put them in the refrigerator then to keep them cool.
It just depends on what kind. Now, cool dark means, as far as I know, somewhere around 70 degrees or so, just room temperature. As long as they're dry and cool and they're not getting any light, they'll probably do okay. But I will give a link to a plant manual at the end that hopefully can give you some more information, but that sort of information is online as well. Any plant that you want to store, just whatever it is, you can pretty much go online and figure out storage methods. There's a whole lot of plants online that give storage methods, but I do cover that in one of my last slides, a good reference manual. Now, as far as the refrigerator, we use that for stratification, and I'll talk about that in a little bit. And that's for more oak seeds. Those are for seeds that just need to be stratified over the winter. But the temperatures for that, you want to keep those plants at about 34 degrees, or those seeds at 34 degrees. And for some seeds at 35 degrees for that long, it may not work so well. We're just trying to mimic the winter, not a full year. And so I'm not sure. With milkweed seed, we have to do the same thing. And I'll talk about that a little bit. But as far as trying to control something for a full year, I don't know about that. But it surely knows that. I've gotten information off the internet for how to do it with trees.
Okay, thank you very much.
And
Sure.
Thank you, Laurie. Also, if you need seeds from certain trees, sending that out through Alan Wild would be one way to do it. Like I have red oak, and if that's something you need, that kind of thing. Thank you.
Okay.
There are no more questions. Thank you.
Okay. So what environmental factors control germination? We just talked about all the internal factors, what's going on inside the plant or inside the seed, those hormones, those proteins, everything in there. But what's going on the outside? Now, primary external or environmental factors controlling seed germination are temperature, water, oxygen, light, chemicals from other plants, chemical from the air, and then the interaction of all of that above. And if you look at this picture right here, we have seed formation, and then there's the environmental and the genotype effects. So the genotype, what's already inside the seed, but then there's the environment. And some seeds automatically are non-dormant and go straight into planting themselves. Other seeds, they get to this point, and if there's enough light temperature, they after ripen appropriately, they automatically germinate themselves. Now imagine that's that green ash that can automatically plant itself. But if there's not enough oxygen, there's not enough of the right things, then it can go dormant. And then when it does get enough oxygen and light, everything's right, then it goes non-dormant and it can go straight into a seed. Some seeds go directly this way. Some seeds go directly this way. And some seeds are flexible and can go this way. And so, seeds have evolved so that they germinate when things are right. If they didn't have this programming inside them, then they wouldn't be able to handle all those environmental stressors. And also the seeds that are much larger, your oak seeds, your walnuts, those larger seeds, they have a lot more inside them to handle those environmental stressors. For example, I work in a bottomland hardwood forest. My trees are generally deciduous, and I work with a lot of oaks. And oaks, I love growing oaks because they're so forgiving. They pop out, and as long as I can give them water, they're going to do okay. And if something happens and a small freeze comes in, or there's not enough water, they're getting too much sunlight or too little sunlight, they kind of die back and they let me know things aren't going well, and then I fix it and they come back alive. They come back to life, and that's how I can have 500 plants at the end of this growing season, and they're ready to plant in Arbor Day time in November. But other plants, not so forgiving. A good example is pine. Pine, they're little tiny seeds, and they need light immediately when they grow, and if things aren't right, they immediately die back and they do not do well. And so larger seeds are a lot more forgiving and can handle a lot more. Smaller seeds, not so well. Now let's start with water, because generally we think just add water and things go. Well, when you add water to a seed, a lot of things start. First of all, a lot of times you find that the seed may have a little pointy end, and in that pointy end is where the water starts to go in. It starts to seep in. And we think of seeds and they're desiccated, they're small. Think of a raisin, you know. You start adding water and it starts to bloat and swell. Now, when a seed swells, it swells from one end and that end starts to get fatter, and then eventually the whole seed will get fatter. It's not that the whole thing suddenly just grows all at once. It's a slow sponge, slowly getting fatter at this end, and then the whole thing goes. And so it's quite interesting that it doesn't all just whoop, turn into a ball all at once. And this is a slow process, and it has to be a slow process because slowly physiological processes need to start. And then there's a special word that we need to cover, and that's respiration. Put your hand in front of your face. And in a post-COVID world, this is a weird, creepy thing that we do now, but you know, just breathe into your hand. There's a lot of things going on. You feel heat, you feel air, and to be able to do that one thing, we have to have water in our bodies. We have to have oxygen in our bodies. We have to have heat. We have to have all these enzymes doing all of these things in our bodies. And that's respiration. And seeds do it too. Seeds don't use carbon dioxide, they use oxygen. And with water, the water is slowly going in, and if the water is not fully there, everything stops. And so water's slowly seeping in, and if the seed gets all of the water it needs, things are going to happen. But what if there's moisture stress and drought issues? So by moisture stress, I'm more talking about flooding, as that question was earlier. Too much flooding, but then there's also drought issues. Well, on bottomland hardwoods, where I work, our forest, we only have an elevation difference from the lowest part of our forest, which is the Trinity River, all the way to our flood bank. Only 12 feet difference. That's not a lot, especially when you're talking about over a mile or even up to 4 miles. Very slow elevational differences from here to here. But the land is going up and down, up and down, up and down, up and down. And so you see all of these changes here. We have areas that dry out, areas that hold water, areas that dry out, areas that hold less water, areas that are higher and drier, areas that still hold a little water. And in all of this, you also have all this shade going on. And bottomland hardwood forests are the most diverse forests that there are. In Northern America, and that is because we have so much topographic differences. Even though it's very small, it could be just a few inches. It could be 1 inch, and it makes all the difference, whether it's going to be a flooded water that's more moist or a drier area that's higher and causes more water issues. And so, depending on what your life history is as a seed, this could be a good thing or this could be a bad thing, but it carves out niches for plants. And so cypress trees and tupelo gum, these guys grow in very, very wet areas, very flooded areas, things like this, old oxbows. And their seeds actually float. And these seeds have to float until they hit land, so they'll hit the edge of the swamp or an area that is wetter but still drier, and they will germinate there. They do not germinate underwater. However, eastern cottonwood, black willow, American sycamore, these are all plants I find right here at the edge of the river. If I'm in a forest I'm unfamiliar with, someone dropped me in our bottomland hardwood forest and said, walk one direction, let me know what you find. Find the river. I would look for these plants. If I find these plants, I know I'm very close to the river. And these guys can actually grow and they can actually germinate underwater, which is ironic because the levee of the Trinity River is actually about this high in our river system. Or no, more like this high. It's as high as the high ridge where live oak and loblolly pine would grow. But these guys, they can tolerate these drought conditions that happen here, but they can also germinate underwater. Whereas cypress, they germinate on land and tolerate flooding very well, but not drought. And so, you know, these trees from here to here have a very different growing history, and that's determined by water, flood and drought. How do they do? A green ash, they can handle flooding just fine for a little bit, so can box elder, but after a certain point, they drown. They drown. And that's the case for many other seeds. But oaks, they all handle flooding very well, and that's why they persist in bottomland hardwoods. But loblolly pine does not persist very well. It can handle wet areas that are wet temporarily, but not flooded for long periods of time. And they don't like soggy, flooded wet feet. They can tolerate wet feet. Now, all other pines do not tolerate flooded feet at all. They need well-drained soil. And so that's how all these plants carve out these niches. And because we have such a diversity based on this little bits of topography, only inches of difference, as I mentioned before, we have all this diversity of plants, which leads to an incredible diversity of amphibians, reptiles, and birds. Also, environmental factors that control germination is temperature. For example, over here, this is the temperature scale over here, and at 35 degrees, which is about 105 degrees, you know, certain plants do just fine germinating, and they can also germinate down here just fine at about, let's see, 70 degrees. This is green ash. Remember, I said it can germinate at a low temperature, and if things aren't right, it goes into a dormancy, and then it comes right back later. Locust, on the other hand, based on soil temperature and moisture, it has a very wide germination rate. Then let's look at red, mulberry, red mulberry. Over here, it germinates at about 85 degrees to 90. And it needs a very warm temperature to germinate. So think spring. So depending on temperatures, you know, plants germinate at different temperatures and at different moisture levels, and finding that right point is where these plants are going to germinate. Now, let's talk about day length, because this is what I found by far the most fascinating of all of the things that I learned making this presentation. Now note, when we plant seeds or when we store seeds, we store them in dark places. And then sometimes when you're planting seeds, say in the prairie, they say dump them on the ground. You don't have to bury them. In fact, don't bury them. You're going to do a disservice. Mow the lawn, set a fire, do something, set fire to the prairie, and then dump all those seeds out there. And you're thinking, but wait, don't they need to be planted? But no, they don't. Whereas other seeds, think you're planting your garden. You set your carrot seeds right on top, but you don't bury them. But then your other seeds, like your peas, you have to bury them. So what's the deal with all of that? Well, some of it may be water, but the other thing is light. And this is just, wow. Now, have any of you ever heard of growing lights? These are very, very particular wavelengths. When we see daylight outside, what we see is the spectrum of the rainbow, but it's all coming together and we see white light. But that spectrum is always there unless we break it with a prism and we can see the rainbow. Plants need red light and they need blue light to grow. This is very important. They do not quite need green light. In fact, if you subject a plant to only green light, it will die. They have to have red and blue lights. And so inside a building, if you're wanting to grow plants, you have to provide red and blue light. You have to provide very specific wavelengths. And I actually got to see this in one of my classes. We walked into a growing room and it was incredible. I walked in and I suddenly came to life. It was very odd. And I thought, wow, it was immediate. I walked in and thought, this feels incredible. I had been in the halogen lights of the classroom and then walked into this growing room and just came to life. And what they had were these red and blue lights in very specific wavelengths. And that makes plants grow without any sunlight at all. And well, when it comes to germination, plants are very dependent upon red light, but not just red light. There is far red light, which is the most furthest part of the spectrum that we see. And then there's red light, which is more to the orange and yellow spectrum that we see. There are plants that do not need light at all. It does not matter, light, dark, it doesn't matter. They're called non-photoblastic. Then you have negative photoblastic, which mean they need dark. They need to be buried. They do not need to see light. In fact, if they see light, they might not even germinate. They need to be buried. Then there's positive photoblastic. And these are the ones that they actually need light, dark, light, dark, light, dark. And they need specific day lengths. And that is very important. Day length is incredibly important, because how are you going to know if it's time to germinate? If the days are really short, what's going on in our world? It is cold. It is winter. What happens when those days start to get longer and longer and longer through time? Spring is here, and we are approaching that spring equinox, when the day lengths are longer and it's warmer outside. And then we know it's time to germinate. And this happens between far red light and the regular red light. And actually, red light is absorbed by a pigment. And this is one of those things that's already in the seed, a pigment. And that light goes through the seed coat and it hits those pigments. And those pigments may actually be on the seed coat. I'm not even sure. But red light, not far red light, the more red light a plant gets, the more that it will go into metabolic activity and mitosis and germination. But if you subject these plants to far red light, you promote dormancy. And I forgot how to explain it, because it's just very confusing to try and explain. But with increasing day length, I believe, with short day length, I think that the plants get far red light when the day is long. But overnight, that far red light, when the days are short and the nights are long, during the night, whatever red light was absorbed during the night, it gets sent back over to the far red light, can actually lose its energy and go down into a regular red light. And once enough red light has accumulated, or you're a nursery and you subject your plant specifically to that wavelength, which, let's go back to that previous slide, this wavelength is 650 to 730 nanometers. If you subject plants to this larger wavelength over here, 730, you will keep your plants in dormancy, your seeds. But if you decide you're going to subject them to 650, they will germinate very well. Look at this, 92% germination. But and that's if they're subjected to just red light. But if you take a red light and then you subject it to far red light, you'll get only 4% germination. Do the same thing, do it in cycles and end with a red light and you get germination. Keep doing that cycle, and then you end it in red light, you get germination. End that cycle in far red light, it doesn't work. And plants have this inside them. They automatically have this inside them. And that red light is what activates gibberellin, which is a metabolic inducing hormone. We'll talk about that a little later. But remember that respiration that we did? Breathe into your hand, oxygen moves through, all these metabolic activities happen in our body. Same thing with theirs. Oxygen is an electron acceptor, or actually it is an acceptor. And when you go into respiration, which is not photosynthesis, but respiration, where you start moving, you make energy. Energy is happening inside the seed. It sets off all of these activities. And when a plant is not getting oxygen because maybe there's too much water in the soil and it's inhibiting oxygen from moving through the soil. Or maybe the seed got planted, but it needs to stay on top of land and it's not getting the oxygen. It's going to be dormant. But once the test of the seed coat is starting to be broken down and oxygen starts leaking in, bam, respiration starts. The seed takes its first breaths and comes to life. And that is when everything, all the inhibitors get inactivated and growth just begins. Now, cypress and tupelo have low oxygen needs, but other seeds have higher oxygen needs. And that's why cypress and tupelo can grow in these soils that are very wet and have low oxygen, whereas other seeds just can't. They don't do as well. Now, talk about seed beds. We just talked about the soil of the cypress. Seed beds are very different. They may have a high litter layer, and that will affect your topsoil environment, and that will affect how much rain and light gets in there. We've already talked about water. We've already talked about what light does, how it affects hormones, and how it affects metabolic processes starting. It also slows how warm that seed bed gets, because remember, we already talked about temperature. Temperature has to be the right temperature for seeds to go. So if you've got a litter layer, you may be affecting all of that. However, remember, there are some seeds that need a dark cycle, a long, dark cycle, and to be able to do their thing. If they get light, they don't do well at all. And then you have your seeds that need to be on mineral soil. A fire needs to pass through, or you need to do a mowing, and those seeds need to get down on there. And that's why a fire is best for working through a prairie. You can go through and mow it, but you're going to create a thatched layer. Fire is by far the best for these types of plants, or for a forest fire, and that's what gets your early successional plants growing. You know, that mineral soil that's right there, that's going to get those seeds that need a high light concentration to go through. Also, if you're a seed that goes on top of mineral soil and the mineral soil is just right, you're going to get lots of sunlight, you're going to get lots of light, you're going to get lots of oxygen, and you're going to get a good contact on that soil, and you're going to get what you need to just suddenly pop out of the ground and grow. And then there's the last thing that affects the environmental factors that control germination, and that's chemicals. Soil salinity. Is there herbicide residues? Is there fertilizer residues? How salty is the soil? What's the pH of the soil? But then also, what plants are surrounding that soil? You may have a plant like black walnut or creosote that produce toxic chemicals to other plants. That's called allelopathy. And that may come off of gases from the plant. That may be gases or chemicals that are exuded by the leaves and then get washed down onto the ground and splashed around. Or it may even be exuded from the roots. And so even the roots have a way to say, stay out of my space. This is my space. And a great example of that is looking at the desert and looking at plants in the desert and seeing how far apart they grow. You can actually see patterns from aerial views. They have wide ranges between them, saying, this is my space, stay out of my space, this is my water, this is my air. And that's because desert soils have very little nutrition. So how do we break seed dormancy? This is my favorite thing because I grow trees, and this may become one of your favorite things because if you collect seeds, it's fascinating that every seed needs different things. So first things you can do is stratification, and this is you have to break seed dormancy with time, temperature, and moisture. And this is mimicking their natural life history. So with an oak seed, let's start with oaks. What is it called? Sweetgum, other deciduous seeds. A lot of them, you take them in a plastic bag and you put moist, nutritious soil in there, but soil that doesn't have a lot of thin soil, but more like leafy litter, so that there's a lot of air in there, a lot of exchange. You take that bag, fill it with soil, fill it with oak, put a little bit of water, not that it'll drown, but just keep the soil moist, put that in the refrigerator, 34 degrees for 6 to 8 weeks is really what oaks need. And that's just above freezing. That is 40 to 34 degrees is what you want. If you freeze that seed, it will die. Do not freeze the seeds. And that is for down here. In the north, it may be different because the ground freezes there. Here it does not. And so by doing that, you give that seed time to stratify itself, for that seed coat to break down, all the after ripening to happen inside, and slowly the tensile strength of the seed coat will break. And it mimics our winter. As we mentioned, you can add red light, 650 nanometers for 8 to 12 hours a day, mimicking spring. And remember, when we're mimicking spring, we're also mimicking that with the light in a forest, in a deciduous forest particularly, all the leaves have fallen. So that red light is coming through to the forest floor. And that additional red light over time, once that pigment has in the plant, the seed has absorbed enough red light, it's going to go. So you're slowly mimicking the forest putting on leaves over time. And that's part of what's going on with that red light with seeds, is that you're mimicking that winter with no leaves, and then slowly, those leaves will come on and those seeds will know what to do. And then you can just straight up add gibberellic acid, ethylene, or cytokinins as hormones, and you can just make those things go. So we know how to break seed dormancy, nurseries do, and there's a book that will have that information that I'll reference at the end. And then also is scarification, and this is what I have learned a lot about in raising seeds. There are different ways that you can break a seed. And that's what this is, scarification. The other was stratification. This is scarification. You take that seed, you can cut it, for example, with an avocado pit. You scar the outside, slice it a little bit, and let oxygen and water in. Now, I've never been successful in that, but I've never really tried, but that's what I've heard. Other seeds, you can just use a nail clipper and bite off the little tip. You can use sulfuric acid. You can get sandpaper and scratch that seed coat, damage it enough to where air and water can get in. What I've had to do, and this is really interesting, this is called serotiny. This is how we all know about forest fires and that they are necessary for forests to grow properly. You subject a forest to fire, a low-grade fire, and the pine cones open up and the seeds come out. Well, how do you do that without a fire? I've recently wanted to learn, how do you grow loblolly? And what you do is you take that pine cone and you put it in your oven for about 45 minutes at 200 degrees, and it works. The pine cone, you get tight pine cones, and then as you subject them to heat, they open up. And then they open up fully, and you can just take that pine cone, flip it over, tap, 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 and all the seeds come out. And it beats trying to break open the pine cone petal by petal and then breaking the seeds from there. Don't do that. I have done that, and that is a bad idea, and it hurts. So use your oven and pop out those seeds and mimic what would normally happen. And here's another fascinating one, redbud. Redbud. I read the directions for redbud, and I thought, this is insane. Take your redbud seeds that have fallen fresh off the tree, put them in boiling water, rolling boiling water for 5 minutes. And then take them out and put them in, just like oaks, a bag with some soil, moist soil, and put all your redbud seeds in there, seal it, leave it in the fridge for 2 months, 6 to 8 weeks, and you will get incredible germination. I was surprised. I had my doubts. How can this be? I am boiling this seed. I'm cooking it. Worked like a charm. Just like pine and fire, redbud grows in those upper sandy areas that can be subjected to fire occasionally. Now, do we have any questions before I go on to the final slides? I know this is a lot of information, but isn't it fascinating? It's fascinating.
Laurie, it is all great information, and I think you're giving us so much information, no one has any questions yet. So maybe we'll go on, and at the conclusion, we will have some questions in chat.
Fascinating. Fabulous. Let's do it. Okay, so let's just get to these last 4 or 5 slides. And we've already talked a lot about this. We've been talking about storage and how do you break dormancy. And so here we are at imbibition. And this is where you drink water, imbibing. Just add water. And we'll cover all of this very quickly because we've pretty much covered it. We're just now talking about the physiology, the mechanics that happen inside the seed, the metabolic processes. So you take that seed and you give it water and it starts to grow. And with that water, those metabolic processes start. And as I've mentioned, depending on the type of seed, the water going into the seed will go at its proper pace for the seed to do its thing. But that internal water, as it goes in, it creates a negative pressure. And so like a straw or capillary action, once water starts going into the seed, it starts to just create this pressure, this vacuum, and the seed will automatically just start. It creates this negative pressure that sucks that water right in. And it goes in through the micropyle. And then once mitochondria get water and that oxygen, respiration, as we talked about earlier. And that, bam, starts all the metabolic processes. Respiration creates this high energy charge. And with all that energy, things just come to life like a factory. And various respiration processes happen. It's not just one. There are actual stages of this. And that's based on morphology. That's based on the type of carbohydrate reserves that we have in there. And it's based also on how quickly that seed coat lets things in. And so some seeds take off instantly and just bam, come out of the ground so quickly. And then others take their time, slow as can be. But with that, respiration is an energy charge. And when you add energy to a situation, when you add ATP, think about when you exercise, and then you just get this burst of energy. It takes a little bit of energy to create even more energy, and that's what's happening. So as that water goes in, it hits the mitochondria, and the enzymes start doing their thing. The factories turn on, and then you start having growth within the seed. And so the growth is going, and as the water finally settles in the seed and it has all the water, the sponge inside the seed is totally soaked, respiration will stabilize and things will go. And then the enzymes that very first started, they'll have done their thing and they kind of slow down. But then another respiration burst happens, because now the mitochondria have created even more enzymes. Growth is happening. And now that these new enzymes are there, they're producing even more growth. Now things are starting to change inside the cell. And also your sugars, your fats, your amino acids, your proteins, they're all being mobilized. They're all moving. And so what's growing, what you knew as a cell, as the seed being nice and fat now, think of that acorn, nice and fatty, it's slowly going, and the storage inside is diminished, and then things start to slow down. But all of this is dependent upon all those factors that we talked about earlier. And those enzymes that I talked about, they're turning over. And I just covered all of this, but it's all of these carbohydrates, starches, lipids, proteins, hemicelluloses, the phosphate. And phosphate is just so, so important. You need to have phosphate for all of these things to happen. And a result of all of these enzymes eating everything is that, and using all of these things, is that those chemicals, those lipid starches, they're all moving around. And now it's like an embryo inside of a human. Things are changing and growing and forming, and now you're going to start having growth. So, we see the outside of the seed, we don't see anything happening, but on the inside, things are happening, things are growing, things are changing. That embryo is growing cotyledons, it's growing its first leaves. And that phosphorus that I mentioned is so incredibly important, because phosphorus is the basis of so many of the protein things that are going on inside of us, and also your cell walls. In humans, phospholipids are in all of our cells. They are the phospholipid bilayers, which all of our cells are dependent. If we don't have phosphorus in our bodies, we die. Our cells can't function. Things can't go inside and outside of our cells. Well, same things with plants. And then also nucleotides, our DNA. is based upon phosphates, which is from phosphorus. And so without that, they can't grow new cells, they can't grow new DNA, they can't grow new RNA. And so without these nucleotides and nucleic acids form the base, nothing happens. And ATP is phosphorus, adenosine triphosphate. That is energy. Our bodies run on energy, and so do plants. They need ATP, and the backbone of ATP is phosphorus. Has to be there. So if the plants don't have enough phosphorus and phosphates, they are not going to do their thing. Nothing is going to go right. And they have to get that phosphorus from their parent material to even have it in the plant. So that's why it's so important that we maintain soil health for all of these reasons, but especially phosphorus. And then for my final slide, then we have seed germination. And so the seed will go ahead and put down its radicle, which is this part right here. That is the root. And that is, all of this, everything that was in that cell, or in that seed, has to grow so that it can finally put down that radicle. And then that radicle will also start to put out its little hairs and pull in more nutrients in. And as the nutrients are still inside that seed and it's getting more nutrients from the ground, then it'll push up its hypocotyl. And then with that hypocotyl, it'll keep going and it'll start to put out nodes, and then it'll put out its leaves. And with the leaves getting sunlight, then it'll get even more of what it needs, it's getting oxygen from down here, actually, from the roots, from the soil. But it'll get the carbon dioxide, the sunlight, and with the water, then we have our whole tree growing. Now this is an oak, and the cotyledons are actually down here the whole time. That is why their seed is so large. So these are not the cotyledons. These are not the first true leaves of the plant. The first true leaves of the plant are actually down here. And then it puts up these leaves over here that start to get, get energy from the sky. So that is hypogeous germination. Then you got epigeous germination. And this is more like a pine or a prairie seed, where the seed is a very small seed. It's really small. And some of these are really minuscule, as you've seen. It finally puts down its roots, and it's putting everything. It's got everything into putting up this guy up here. And, the roots are doing what they need to do. And then the first leaves, these are the first true leaves. This is the factory right here. And if these don't get out of the ground and get sun immediately and get what they need, then this plant will just die. However, over here, because the hypocotyls have so much starches and energy inside them, and they stay below ground, and there's so much stored in there, if these die, it has about 3 chances that it can come back up and put out new leaves, because its first true leaves are down here. It can try putting up new leaves, and this can even grow in shade. And these are evolved so that even just a little bit of sun, dappled sunlight, will do the job. It doesn't even have to get direct dappled sunlight, just enough light coming through the forest canopy, and these will do just fine. But pine and other flowers, if they don't get 100% light like they need, boom, they crash, they die. And so that is the secret life of seeds. So much going on in there that we just have no idea. And I don't know why my slides are not changing. There we go. That is my final slide right there. And this is the Woody Plant Seed Manual. And so unfortunately, this does not cover flowers, but this has everything you could possibly know about woody plants in the United States. And I actually only found this about a month ago. I wish I had known about this before, and I need to find a copy somehow or keep a link to this, because this is where you find that information about how do you stratify, how do you scarify a seed, what are the best ways to store them, collect them, store them, germinate them. But all of that information is on the internet for other plants, I promise, because seed nursery information is in high demand, especially now that we are trying to promote pollinators and trying to promote prairies and and I know that you can find it online. Now, I know this was an incredible wealth of information, just so much information, and I hope I did not give way too much information, but I am okay with Chuck forwarding these slides to everybody, and so that you can reference them and look at them, and you have my permission to go ahead and give this presentation as well to others, because this is just such phenomenal information. And the slides, when you make presentations, you're not supposed to put a lot of words on them and just use bullets, but I prefer to put as much information on my slides as possible so that people can share them and reference them later. Also, it helps if I haven't given the presentation that it kind of got a script right there on screen. So with that, I will take any questions that you may have.
Okay, Laura, we do have a few questions. One,
Okay.
that she can't unmute, so I'll read it for her. Can seeds be initially grown in sterile medium, or do they need a growing mix containing nutrients from the beginning?
I don't know about that, actually. I've never tried to grow seeds in a sterile mix because I always go ahead and just get potting soil. I would say that I wouldn't try doing a sterile mix. I would still go ahead and try using, do sterile mixes have the mycorrhizia in there?
Usually
I
not.
don't know. Okay, I would prefer to grow them in soil. And by sterile mix, does that mean perlite?
I think maybe perlite. I mean, I germinate some milkweed seeds like in vermiculite, which is pretty sterile, and then immediately transfer. So I think that's what perlite, vermiculite, even sand probably would be a kind of a sterile mixture, right?
Yeah, I don't know. I did mention earlier about how when the seed germinates, how is the soil conditions of that? I would say that probably in these seeds that are larger, you could possibly do it in a vermiculite, but I don't hear of anyone doing that. I would definitely grow them on something with nutrients, but that is something I would definitely consult the internet for better information. I typically just buy bags of potting soil, and what I do with my operations, in case anyone's interested, I get a pot, and I'll put 4 seeds in there, and then I'll go ahead and put it outside after I've stratified them and let them do their thing. I find that it's actually much better to stratify them in the refrigerator than out in the wild here, because our winters are so variable in how warm they can be, and also because the squirrels get to them. And I have much better luck putting them in the refrigerator because the squirrels, the nematodes don't get to them. I use a potting soil, so I don't use the soil directly outside, and that way I have less pests getting to my seeds. And once I've potted them, I'll see how many have popped up. And then once they become hardy enough, or I know that the seed, the root is really small, I'll go ahead and use a trowel or 2 and get them out of the ground and then transplant them to different pots. And so they'll all have their own pots. The other thing I did not mention about this in this presentation is that I started this project with maybe 45 acorns one year. And they're just a mix of acorns that I had found on the ground using native seed source. And I went ahead and planted them in the pots and thought I would only have 45 trees to plant because I only planted 45 seeds. But here's the beauty of not having a nursery and just having a backyard. A lot of the seeds will float in, very light seeds, and they will land in your pots. And I have a sycamore tree right in my backyard that puts out gobs and gobs of seeds. And then other seeds that have flown in are green ash and also red maple. And not red maple, but box elder. These seeds come from outside my yard. I don't even see any of these trees near my yard, but they float in with the winds and they plant themselves in my pots. And so I will take all of those seeds and transplant them to different pots, and that's how I'll end up with 500 plants. I go through a lot of potting soil, and I buy potting soil, not garden soil. I learned a lesson with someone donating soil to me this year. And I had a huge crop failure. And that was because the soil, though it was potting soil that they gave me, it had a lot of fine particulates in it, a lot of fine soil matter. And maybe it was because this potting soil had been in storage for so long that the microorganisms inside had broke it down into finer soil. But that fine soil drowned my seeds. They couldn't get enough oxygen. They got too much water, not enough oxygen, and they drowned. And so I prefer to get a potting soil like Miracle-Gro, and then grow my seeds in there, and they do pretty well, outstandingly well, to where the point is, what am I going to do with all these seeds? I've got about 500 seedlings in my backyard right now.
That's a fun problem to have, though. Better than the other, right? Not having any germination.
Yeah, that's what I was worried about too.
Okay, we've got 2 more questions. Mike, would you
Sure.
like to unmute yourself and ask your question? Mike?
Yes, I would like to know if the storage vaults in Norway use any inert gases to preserve their seeds.
That I don't know. That is fascinating. We've donated seeds through Trinity River National Wildlife to Kew Gardens in Britain, and we've contributed to the vaults. I'm not exactly sure where all of the seeds have gone to. People come and pick our seeds specifically for the Millennium Seed Project, and I don't know if they use gases, but what I was told at the time is that they simply use temperature. But they didn't say we don't use gases or anything else. They just said we keep them at a proper temperature and dried condition so that when we need these seeds, we have them. But that's a fascinating question. I should ask my source about that.
Thank you for that. And we've got one more. Peggy Carroll, do you want to unmute yourself and ask Laurie your question?
Hi, Laurie. You mentioned milkweed earlier, and I wonder if you could talk about germination of milkweed seeds.
Well, I am big at failing at the germination of milkweed seeds. I will admit that right now. And maybe part of that was because when I was trying to germinate milkweed, it was 2-3 years ago. And so I didn't know about the light, and I didn't know about the moisture and everything else, and the temperature. And so I was just kind of under the assumption of put milkweed seed in, something will grow. And I had very poor germination. But also the milkweed seed that I was trying to grow was not native to this area. And at the time, I didn't quite understand why the stratification was so important of the milkweed seeds in the refrigerator. And of course, it says that in the package. And so when you buy your milkweed, make sure that you're buying milkweed that's native to this area. It's just, we have not had great luck with milkweed in this area. And in my refuge, perennial or swamp milkweed, Asclepias incarnata, the milkweed that grows the best in our floodplain. But then we also have antelope milkweed growing in our drier areas, in our sandier, drier areas, and it grows natively there. And there's also one other milkweed that I've found, and I can't remember the name right now, but a whole lot of milkweeds grow in this area that I was so surprised at. Because I don't generally find them, but I'm not that well versed in milkweed. But buy your milkweed seed at the right time. That is, don't buy it immediately right when it's planting season. Buy it when you have time, the adequate amount of time to put it in the refrigerator. It has to be in the refrigerator for 6 to 8 weeks. And so when I purchased my milkweed seed the first time, big failure, because I didn't stratify it. I just assumed you put it in the ground. And also, you know, when I was talking about the red light part, something that I found interesting as I was doing the research for this presentation was that some plants only need a very short light cycle, and by minutes, very, very short. And so now when I plant my seeds, when I'm planting my garden, I go ahead and set those seeds out in the sun for a while, give them that boost before I go ahead and put them in the ground or on the ground, because naturally, if your pea plant or whatever plant is dropping that seed, it's going to drop it on the ground. It's going to get sunlight, at least for some period, before the other leaves fall on top of it or cover it up with soils. As rains come, they splash soil and move it around. And so that's how things also become naturally buried. And so don't be afraid to give your seeds some light. Now, as far as milkweed, also just making sure not to drown them. Like I said, I've had very poor success with milkweed, but there is the Monarch joint venture, and they have a lot of information on growing milkweeds, and I would try going with them. But learning about growing milkweeds, I'd say 3 months in advance of the planting season is definitely the way to go. And then last thing is making sure you get your seed from a good supplier or going directly to the plant itself. I think somewhere soon is about the time when antelope milkweed starts dropping its seeds, if not already. And picking that seed at the right time. Wait for those seeds to start to darken and desiccate and drop out of the plant. Don't go pulling them off the plant before it's ready. Is also going to be a big part of it. But definitely look up on the internet the best ways of doing all of these things, because there's people who definitely know more than I do, and that's just scraping the surface of the information of these plants.
Thank you, Laurie, for that. And
I wish I had better answers.
Oh no, that's okay. I'm a milkweed seedaholic myself, so that is my one. I'm a seed collector and a seed grower. I have loved this program. I don't think I could have read enough books to get the knowledge that I've gotten in this program today. It's just amazing. And
thank you.
As far as the milkweed, it does need, as you said, the cold stratification. It absolutely has to have it. And the fresher milkweed and the more local milkweed seeds, the better germination you're going to get with it. I appreciate everything. We've been asked for your email address. Could you put it in chat so if people have other questions that they could send you the questions personally with your email address? Because I don't think we had it in any of the slides. And we have all kinds of comments I think we're going to save the chat to all kinds of comments about how wonderful your presentation was and how helpful. There's a lot of seed collectors here. I know that.
Great. All right. There it is.
Thank you very much.
You are more than welcome. And I hope everybody learned a little something and didn't drown in all the information.
No, a lot. You got us at all different levels.
Great.
So thank you for that.